Okay, so alternatively,
I'm going to speak with Dr. Hussein. He's a fantastic professor, part-time here at the new school, and also he's at the
Zuckerman Institute, yes.
At Columbia University. But then you can say more. And
Yeah.
we're really excited to have him here and to do something very, very new to try to connect neuroscience to the principles of AI. And I think you can explain better the same that
Sure.
is where his field of expertise is. Yeah, that's it. So,
All right.
really excited with that.
Yeah, thank you. The yeah, as you said, the area that I've been working on, I think it's been rebranded to some extent in the past few years to NeuroAI. So that's a term a lot of people use now. So I'm going to try to give you some intro, some background on neuro, and talk a little bit about AI and how they connect. I'm going to use examples from my own work, just a case of studies that where do these two fields meet. We're not going to get into design. We'll leave that for later. Maybe in a semester or so, we'll have to say more about how it all connects to design. So I'm going to start talking about the nervous system. I don't know how I'm not going to assume any backgrounds, but Andrea told me that don't hold back. Like you could just go as deep as you want, and then people hopefully follow. If not, this is going to be recorded and you could watch it later. So um, this is a schematic of the nervous system where we have the brain, and it's connected to the brainstem, and then the nerves can extend to the hands, the limbs, the legs, and all parts of our body. So let's see. So the brain is sitting in the skull. The brain is sitting in the skull up here. This is a very general schematic of it. You probably have seen an image like this before. It shows the four lobes. You heard of some of these lobes, maybe frontal lobe or occipital lobe in the back. This is cerebellum, and this is the brain stem. What's on top, this is what we call the neocortex. This is evolutionary new. If you go to our ancestors, like lizards and frogs and whatnot, they don't have the neocortex. This is something that evolved later. No, this is the entirety. So if you cut the brain right from down the middle, so imagine I have a right side and the left side of my brain, and I cut it down the middle, and you're looking at the right side of it. And this white area would be the nerves that were connecting the right and left side of the brain together. This whole folded thing on top, this is what you see at these lobes. The whole folded thing is the neocortex. So in reality, the actual nerve and nerve cells that I'm going to get into are really at the surface of this. So if you took this out and spread it out, it would be like a dinner napkin. But because it's kind of jammed into a skull, it's going to be folded like that. These folds, we call them, each of them would be, when it protrudes, it's a gyrus, and it folds in, it's a sulcus. So let's say now you have the brain looking in the left direction, and if you take a cut like this, you would see that the actual, as I said, the actual nerves, what we call the gray matter, is on the surface. This is on the surface of the cortex, and it folds in and out. All of these are basically connections between them, and we're going to get deeper into that. But as I said, this whole area that is sitting on the surface, we call that the neocortex. This is you may have an idea now. The person is looking to the left, similar to here. And you're looking at this left hemisphere, right? Like the stance is the stance that I have right now, and you're looking at the left side of my brain. And this side has 4 lobes, and the 4 lobes repeat on the other side too. So these are in both hemispheres. Yeah, the takeaway from this is that, well, just the general architecture. When you look at, macroscopic view of the brain, this looks like this. A lot of the things that I'm going to say today, it's going to be basically limited to the cortex and the processing that happens in the cortex, because a lot of the high-level cognitive behavior that we have and capabilities really come from cortex, right? They don't exist in more primitive animals. So if we put the tissue under a microscope, and imagine you're taking the cut, in this case, would be, let's say you cut this in this direction. So in the depth of the surface, so imagine I said it's a dinner napkin. If you cut it and then you look at it under a microscope, it would look something like this. This is a drawing from Santiago Ramon y Cajal. He was one of the pioneers in this field and drew these things 120 some years ago. And he would stain these cells, he would inject them with some dye, and then he would go and draw them, look at them under a microscope and draw them. What we know, like roughly, is that in 1 cubic millimeter of the brain, there are around 10,000 neurons, and there are 3 kilometers of wires, so about like 2 miles of wires, if that means anything to you. But it means that it's really packed. So these cell bodies are really small, and they are really packed in this small millimeter cube of the brain. These are what we call pyramidal cells. These are the most common cells in the cortex. So if you look at one of them, what is the architecture? What is the structure? Well, the structure is that, like any cell in the brain, well, any cell in the body, you'll have a cell body, you have a soma, where all the mechanics of keeping the cell alive and the nucleus and the gene expressions would be maintained there. The big difference that neurons have with all the other cells in the body is that they have dendrites and they have axons. And these dendrites and axons are places where they could receive information, and they also pass on information. So how do they do that? Well, cells have something that we call action potential. Action potential is the fact that the cell becomes excited, and I'll explain what that means. When the cell becomes excited, the voltage inside of the cell really goes up. And when that goes up, the cell kind of metaphorically explodes, and that positive ion or potential travels down the axon. And you could actually record that. You could actually listen to what that would look like if you put an electrode next to the cell body and listen to the cell, like you actually record what the potential is. So when the action potential, when the action potential reaches the end of these nerve endings, basically the axon terminals, it releases what we call neurotransmitters. And you've all maybe heard of neurotransmitters, right? What are some example neurotransmitters?
Okay, that's fine. What would be that?
Okay, yeah. So, for example, when you take antidepressants, what neurotransmitter involves most of antidepressants? So SSRI stands for what?
Serotonin.
So serotonin reuptake. So serotonin is a neurotransmitter, right? So what does serotonin reuptake inhibitor do? Well, if there is, let me get to that later. But imagine serotonin is a neurotransmitter, and all of these neurotransmitters are released at these nerve endings. So at the nerve, at these axon, at the end of every axon, right, there is a synapse. And the synapse is where one presynaptic neuron connects to a postsynaptic neuron. And this is where neurotransmitters are released. And when neurotransmitters are released, they could impact the postsynaptic cell. This is a way of communicating. If a neurotransmitter is excitatory, and that's the terminology that we use, like serotonin, like dopamine, these are excitatory neurotransmitters. What they do is that they excite the postsynaptic cell. They make the postsynaptic cells also be active to have an action potential. If they are inhibitory neurotransmitters, like GABA, for example, they inhibit the postsynaptic cell. Now, there is the mechanics of ion channels and how they go about exciting or inhibiting. We're not going to get into that. But know that what neurotransmitters do is that they mediate communication between neurons. So serotonin, I said, is an excitatory neurotransmitter. It means that when one cell releases serotonin, it makes the postsynaptic cell more excitable. It makes it fire more. It makes it be more active. Now, when a cell releases these things, they have to reuptake them. They have to go to their baseline and then to be able to release more. So there's a serotonin reuptake. Now, if you take a serotonin reuptake inhibitor, what it does is that it leaves a lot of serotonin between cells. And that would cause your cells to be more excitable or no? What would be the effect on the cells? If I take an SSRI, which is serotonin reuptake inhibitor, it means that there would be a lot more serotonin that are not being taken back. So they just exist between the cells. And they exist between the cells, they make a lot of your cells more excitable. And that's what their effect is. That they make cell communication stronger, and then they make the cells a lot more excitable. So the takeaway here being that cells in the brain communicate with one another. It's not like cells in the body also communicate in other ways, in electric fields and all sorts of complex ways. But in the brain, the way they communicate with one another is through the release of these neurotransmitters. So if a cell is really active, in a way, it could signal to the rest of the brain that, oh, I've detected this, or this is something that I know about. So I'm going to jump into talking about circuits. And again, we're going really fast over this thing. So just bear with me. So if I say cells can communicate with one another, what does that give me? Well, that gives me computation. They could signal different things. So for example, if I have this primitive circuit, as we call it, in the brain somewhere, and these, imagine, are photoreceptors, that if there is light touching each of them, they're going to be active. And imagine these are excitatory connections, and these are inhibitory connections. Well, it means that if there is light hitting this part of the wall, this part of your eye, let's say these are in the eye, they're going to excite this cell. It's going to increase its activity. And if this is active, it's also exciting this one, and it's going to make this active. So if there is a lot of light here, this cell is going to be really active. If there is light here, it's going to activate this, it's going to activate that, but this is going to inhibit this. So imagine, I'm just showing you a very simple example of how you could imagine this was a kit, a toy kit that you were connecting with one another, and all you had were connections that could be excitatory or inhibitory. And you are taking this kit and you're saying, okay, I know that this is a light sensor. I'm going to connect it to this other kit that is excitatory, this unit is inhibitory. So if I wanted to create something that would behave, if I wanted to have a behavior from this unit that would be really active if light was in the center, and it would not be active if light was in the surround, I could connect that kit this way. So what I'm trying to give you, I'm trying to give you a mental model of how connecting neurons to one another can help you detect the stuff, can help you, for example, process light information in different ways. Does this make sense? This is a lot. As I said, I'm going fast through these things, but it's going to make sense why you have to think about it this way. This is something that happens in the brain. So everything, where are all the knowledge of the world and experiences that you have and let's say traumas that you experience, where are these stored in the brain?
The hippocampus.
Hippocampus is an area that is involved in memory formation. But where is the memory itself stored? You want to take a step? What is in the brain? So let's say I said, this is what brain looks like under a microscope. What do you see here? What's that? Nerves, right? There are connections between these units. And these units are cells, right? So they could be active, each cell could be active or not, and there are connections between them. So everything, there's the only things that are in the brain really are activation of cells, that how fast they could fire, and the connections between them. So if you have a memory and let's say word knowledge, where could this information be stored?
Or like that specific connections only lit up with that.
Yeah, so there's only connections in the brain. So anything that you know in a network, in a neural network, everything that you know, everything that you experience, it would have to be part of those connections. Those connections change as part of a learning process. And everything that you could do has to come from those connections. So what is a neural network that we use a lot in our lives? What's that? Neural network? What's a neural network that we use a lot in our lives?
Go large. Like ChatGPT.
Yeah, ChatGPT is a big neural network, right? So I show an example of a neural network here. This is a neural network, and it has 7 neurons in it, and it has, let's say, 2, 3, right? 7, 8, 9, 10, 11, 12 connections. How many connections are, let's say, in ChatGPT 4.5? Somebody take a guess. How many connections do we have? So let's say in the brain, the estimate is that we have 100 billion neurons and we have 100 trillion connections in the brain, right? You saw how dense that is, right? Under a microscope, you could just detect the cells and you could detect the connections between them. And the estimate is that there are around 100 trillion connections. How many connections do you think something like GPT 4.5 has?
I guess 5 trillion.
5 trillion. That's a good guess, right? It's in trillions. We don't know the exact number. They don't release it, but it's in trillions. That GPT 4.5 has trillions of connections in it. It has hundreds of layers. Here I'm showing you, for example, 2 layers of a network. That one layer is doing something, it's processing information, it has a pattern of activation, and that pattern of activation gets passed on to the next layer. So this is how, I'm going to talk about language models a little later, but the idea is, the core idea is the same, that you have units, the units have a set of activation, that activation gets transformed through the weights to another set of activation, and then that gets transformed to another set of activation. So everything that happens in the brain has to do with the transformation of activation through these weights. And those weights are where the word knowledge is. So when you tell ChatGPT, for example, okay, what is the capital of New York? I used the example in the class the other day, and it tells you Albany. Well, capital of New York is just set of, ones and zeros, goes into the network, and then that gets, because of the weights that the system has, eventually gives you an output. But it's all about these weights. This is a much simpler network, and what it does, it responds to something like this. So if you have a pattern of light that looks like this, so if there's a point of light somewhere in the world, this cell would be really, really active. And why would anything in the brain function a certain way? Is because it's connected a certain way, right? Every structure gives rise to function. That's the point I'm trying to make. So if a neuron is behaving, or if the brain area is behaving in a certain way, it's all because it's connected in a certain way, because there's nothing else, right? So when I was talking about the different lobes, for example, the visual response is all in the back of the brain. And the reason is that the back of the brain is connected directly to the eye. So if you're connected to the eye, you're going to become a visual, module, because that's the input you're getting. So because you're connected in a certain way, you're going to have a certain function. So again, going fast here, if I have another unit that is connected to units that are selective for detecting points of light, well, I could become selective for maybe an elongated bar, right? So being selective for something, it means that when that thing is in your receptive field, you're going to be responding to it very strong. That's all that means. So if I say there is an area in the brain that is face selective, it means that whenever you're looking at a face, that area is really active. Okay, so for example, if a cell is really selective for vertical bars, it would be really active. This shows how active the cell is, how many action potential it has. I wanted to show a video of action potential, so it gives you some intuition of what I mean.
And if I'm not saying, so
Yes.
that's why, for instance, they show images to people and put in an fMRI, because you can see if you show a face, for instance, what area is going to be activated, right?
Yeah. So a lot of time, it depends on the experimental paradigm. When it's in neurophysiology, okay, that's good. We have sound. When it's a neurophysiology approach, it would look something like this. This is an animal study. I'm going to show experiments that are done with cats, but in this case, it's a macaque monkey. You put an electrode into their brain. These are all done in humane ways, right? They're under anesthesia, they get the surgery done. You put an electrode in their brain and you're recording from a specific area, or in this case, you're recording from a specific cell. And then you flash light in the environment and see which part of the environment they respond to, and you call that low. That part is the receptive field of this specific cell. And then you could actually play around with the light and be like, what is the specific light pattern that this cell responds to? When you're listening to a single cell, you could just connect that to an amplifier and then either see it in an oscilloscope, or you could actually listen to it if you connect it to a speaker. So I'm showing you, for example, these people who did can you all hear it? Okay, so each of those little sounds is an action potential. It shows that the cell kind of exploded and then it went back to normal, exploded again, exploded again. And if you hear too many of them, it means that the cell's firing rate is really high. You see, so they're saying, okay, it looks like when the light is in this part of the environment, the cell is really going crazy.
What is crazy, right? Energy?
No, it has a lot of action potential, which in chemical term, it means that there are a lot of positive ion rushing into the cell, and the potential goes up, and then it releases neurotransmitters at its axons. That's what it means. Chemically means that. And then it goes to a normal level, it goes back, there's a refractory period, and then it does it again. It does it again. Each of those events could only last 1 millisecond. So the cell could potentially do this hundreds of times per second. And the chemical effect of that is that it releases neurotransmitters. And releasing neurotransmitters means that it impacts some other cells. And those cells impact some other cells. So there's a cascade, and I'm going to get to it. So that area is excitatory, but they're also mapping an area that seems to be inhibitory. When there is a light right next to it, you could see that the cell goes quiet. So this area seems to make the cell go even below it. So cells are not dead. If they're not dead, they have maybe a spike every once in a while. But when you put light in certain area, it really makes them go crazy firing, or it makes them go really quiet. So there's an excitatory and inhibitory area. The effect of that is that this cell is detecting edges in this part of the world. So if there is a contrast between light, that's an edge, right? Every edge in the world, it means that there is a difference in contrast or the color at that point. And that contrast gives you the edge. And if the cell can detect that contrast, then it can detect an edge in the environment. So I just wanted to get an idea of what it means when you listen to a cell, this is how they behave. So there are 100 billions of these cells, and you just listen to one of them as you flash light in the environment. So the study that I showed you is from Hubel and Wiesel. They did a lot of these seminal work in the late 50s or late 40s, 50s, and 60s, and they won a Nobel Prize in the 81 for their work. Hubel is that, but Thorsten Wiesel is still alive and he's like 100 years old. So with this approach, what they did is that they basically they put the electrodes in one part of the brain, and then they flash lights and then see what this runs with. They move it a little bit, they move it a little bit. So they did so many experiments with these single cells, just looking at behavior, and they saw, well, it looks like, and this is tissue. This is the figure that I showed you earlier. This is just part of the cortex. And I said you could flatten it out. You could see that units that are close to each other respond to similar stimuli in the environment. So if this is responsive to this orientation, if you change the light a little bit, it stimulates the tissue next to it. And interestingly, you could see that information from one eye and the other eye are next to each other in the brain. So let's say there is information coming from that, wooden pieces there, coming to my two eyes. One of them goes to the left, both of them go to the left hemisphere because information gets transformed to the left hemisphere, but they end up in the same place next to each other in the brain. Why would you want information from two eyes to be close to each other in the brain? What would that give you? This has nothing to do with the brain. Why would you want information from the two eyes next to each other? What does that get you?
Dimension.
Dimension. What does that mean?
You don't have view, or you have like wired field of vision.
You were closer, right?
Yeah.
So you would get depth perception, right? A lot of time depth perception, if I'm looking at my finger, there's a disparity between the image on my two eyes. And if I have those two images next to each other in the brain, that disparity tells me what is the depth of this thing, right? I could get depth perception just by comparing the response that comes from the two eyes. So a little bit of anatomy of the visual system. As I said, when you look at the environment and you have your two eyes, the right and the left eye, the right visual field would project into the left side of the eye. So meaning that if I'm looking at the world and anything to the right of my hand would have to project to the left side of my right eye and left eye, right? Because there are lenses. Okay, I didn't get into this, but there are lenses in front of the eye. So if there are lenses there, they just, right? What do they do to the light? They flip the light. So light that comes from this part of the world goes to the left side, right? And the stuff that comes from the right side of the world goes to this side. There is an area in between that you could imagine there's an area here where you're not registering any visual input. And this area is called the blind spot. And I could show you how to find it. So let's say let's do this. If you all close your left eye, okay? If you all close your left eye and then put your left finger up and then move your right finger. Now keep looking at the index of your left finger and then keep moving your right finger toward it. There is a moment when the tip of the right finger disappears. Do you all get that? So what are we doing? So left eye is closed, left index finger is up, and you're looking with your right eye to the left index finger. Now move the right finger towards it, and there's a point that the tip of the right finger disappears. You see this? Is everyone getting this? Yeah? You could try it later if you, but no?
Yeah.
Yeah, okay. So that's where there's a spot in the eye where the information is not being registered because that's where the nerves leave the eye. And at that moment, there are no photoreceptors. But because those are at different spots for the two eyes, when you have both eyes open, you wouldn't really know that there is a blind spot. So the information from the right side of the world gets all transformed to what we call primary visual cortex in the back of the brain. So this is where the visual processing, a lot of the visual processing takes place. This is a cut of the brain in this direction. So imagine at the eye level, you're cutting the brain. And when you do that, all the gray matter would be at the surface of the cortex again, as we talked about it. These are all just connections. And these are the nerves that go from LGN to the back of the brain. And this is a crossover, optic chiasm, where you take information that belong to the right visual field because they come from two different eyes and take them to the back of the brain. Okay, so we could take this story of cascade of information and study it in the brain. We're saying, well, information comes through the retina. These are in the retina, there are a lot of photoreceptors, right? They respond to the light in the environment. And those photoreceptors are connected to other cells. And I showed you a sample behavior of a cell in V1 where it's responding to different orientations. And the way people now think about object recognition is that we are building complexity as we go deeper into the brain. So there are areas in the brain that are like phase selective. How do they become phase selective? Well, this is the dominant view right now in the field, where let's say you have an object. In the early visual areas, you're detecting edges. So this cell says, okay, this cell says I detected an edge here. This cell says I detected an edge here. And keep in mind, the entire visual world is tiled because the visual information is projected to the back of the brain. And there are orientation detection in every single place. So in a way, imagine it's a sketch pad in the back of the brain. And this sketch pad tells you orientation. So in this part of the world, there's only these orientations. In this part of the world, there are only these orientations. So all of these orientations would be detected. Now, if there is these two orientations in this part of the world together, maybe you detect then a longer, orientation there, a bar there. Maybe in the next level, you detect, well, these two orientations form an edge. So you detect an edge. Detect an edge means that there's a cell that responds to edges in this part of the world. And then you go up the hierarchy, maybe those edges come together, they form a shape. And those shapes come together in order to form a face, for example. So the way you get a cell responding to faces is through a cascade of these detections that happen in an earlier layer. Those combine to become deeper and deeper and more complex processing. Now imagine something like this happens in language processing and happens in auditory processing, where you have maybe frequencies are detected initially. Those frequencies come together, form small phonemes, and then those come together, they form sounds and words and so on. So these sorts of thinking about brain processing as a cascade of hierarchical processing with bigger and bigger receptive field and more complex features is really dominant. I wanted to show you a demo, but I have it. Let's see if I can find it.
This has a lot to do with gestalt in design.
What?
Gestalt. You study that here, the visual principles of gestalt and information theory. There's a lot to do with that. How to detect edges, and then you have the background and the foreground. Yeah. So all this. Like the
NeuroAI.com's intuitive talent management platform. You have the power, not IT, not admins. You are in charge.
So this is an experiment. I just want to show you how fast object recognition is. So, and why people think this is just a feed-forward process, right? They're saying like information comes through your eye and then it goes into the back of the brain and just projects forward in the brain. And they want to know, like, why do people think it's so fast? Part of the reason is that there are these experiments, rapid serial visual presentation, or RSVP. And in this example, your task is to report whether you see Buzz Lightyear. You know who that is, right? This is the character from Toy Story, right? Yeah. So, and let's say when you detect it, say Buzz. Nothing, right? Yeah, not yet. Okay. Buzz, right? Okay, so imagine these images are flashing maybe for 100 milliseconds. So that's really short. But we're able to detect really complex things about that, right? So people are like, if I can detect, and they're saying, well, there is axon conductance delay. Like if it takes, let's say, 5 to 10 milliseconds for information to go from one layer to another, then the fact that I'm able to detect something. So let's say, oh, from the eye to LGN to primary visual cortex to the next V1, V2, like up. So it's like, it looks like even in the first pass that information gets through, you're able to detect something or recognize something. So that's the idea that what people think they're feed forward. Feed forward means, I'll tell you what feed forward means right now. Feed forward means that the information is just feeding forward, right? So a lot of time you say, okay, the way I recognize things has a lot to do with my expectation and appraising what the situation is and like all of those top-down and knowledge and stuff like that. So feed forward is in contrast with feedback. And a lot of people think, well, the way I recognize things is through an idea that I have that feeds back to the earlier areas, like some of the generative models that Andrea, you know about. But the idea of feed forward is that the information is just feeding into the brain and just moving forward in the way that I described it. So some cells are active, they release neurotransmitters, some other cells become active, some other cells become active, and just feeding forward.
Because if it was feeding back, what would it look like?
I'll talk about it a little bit later. So I want to use the example of face perception and introduce you to some concepts there. We're very good at face perception. We've seen so many faces, right, in our lives. But it's still, we're really good at identifying just a single person or a single face, right? You all know who this is, right? I don't know, people are younger here, but I don't know. Who is this? Clinton, right? And this is Saddam, yeah? And
This is the, from this one, the, I know.
you don't know who that is?
No.
It's, yeah, it's Nixon. Nixon. And this is, right? So it's interesting how good we are at this task, right? These are very blurry faces of people that a lot of them, you know, died a long time ago. And a lineup of people who it didn't age well. It was great in the 90s, maybe early 90s, but the idea is that how do we do this, right? And I'm going to show you recordings of the brain from some higher visual areas. This is not anymore the V1 area, but this is from areas that are generally face selective. So we're looking at a bunch of cells and they're ordered like here. And we're looking at the responses, the firing rate of the cells in response to a bunch of images. So you have responses to human faces, you have responses to monkey faces, and responses to non-face objects. So it's the same neurophysiology paradigm that I described earlier. The difference being the recording is not in the primary visual area, but it's a few layers higher up in the infratemporal cortex, which is right down here, for example. You could see that these cells are, and the color coding is that if it's brighter, it means that it's more active. So, and these are ordered, so the more face selective cells are at the bottom. So what you see here, that these cells are really active, for example, for human faces and monkey faces, and not so active for objects. And different cells have different properties. The point being that it's not that a single cell represents this face. It's not that the activity of a single cell that tells you properties of this face or properties of that face or identity of that face, right? Everything that we experience would have to be a population of cells that are active at the same time, right? Because there are so many of them and one cell is not going to signal anything. So when we perceive something, it's about a population of, let's say, millions of neurons being active at the same time. I mean, when you see a picture of your grandmother, it's a population of cells that are active that signal that, oh, this is my grandmother, or that I've seen this face before, or this is, you know, Bill Clinton or someone else. So this is a population, and we're showing here, for example, 500 some cells. And for this specific phase, this first phase, this population, however they're active, the ratio between different cells among these, let's say 500, is signaling in the brain that this is this phase, that signaling the properties of this phase. Yeah? So we call this population code. And I want to say there could be an alternative. The alternative being, well, I'm representing each phase by the activity of a single cell. So I'll use an example. Let's say you color coding your friends in your phone book, for example. and you give maybe Bill red, you give Samantha green, you give Roger blue, and that's all the color pens that you have. Well, how are you going to do with grace, right? You run out of color, right? You can't really give people just individual colors. That's what we call very specific code. Now, alternatively, you could represent Bill in your phone book or whatever with, let's say, a little bit of red, a lot of red, a little bit of green, and very little blue. Does that make sense? What I mean is that you're using the ratio between these different colors as a way of representing. So this is not Bill. This is just a representation of Bill. Does that make sense to everyone? When we say something is represented in the brain, you're using a different coding system to signal that this refers to Bill. Do you want to say something?
No, think about like the Photoshop scene, or something like that, that of colors in which you have a code that representation of, and we had a conceptual model that's what Hussein calls is like mental model that we saw last class that he touched on.
Yeah. So
This is like an overall rule for like all perception.
yeah, right. So everything in the brain would have to be. So for example, as I said, I'm showing the response. So each column here is one image that is the monkey is looking at. So this, for example, this first one is they're looking at this face. And this is the activity of 500, that's 520 maybe cells. So it's the ratio between these 500 cells that signals to you, well, this is that face. Now, it's a lot, many more cells, but in this case, they recorded 500 of them, for example. So it's not that, you don't expect that when I show you this face, only one cell in the brain is active and everything is quiet, right? That's not how it signals. The signaling in the brain works through a population activity, and the ratio among those population activity signals with it. Does that make sense? So when I say a representation, this is a representation of Bill, the ratio between these three colors, right? I'm coding Bill in a certain way in my phone book. I'm coding Samantha as a little red, a lot blue, and somewhere in between green. I don't know what colors I use, but the colors match across. I'm coding Roger or representing Roger at these combination of colors. And I'm representing Grace as this combination of colors. Now I could have more and more people added to my phone book, right? There's no limit anymore because I'm using a ratio of these things. And if I'm using a ratio of these colors, I could have as many people as I want added. So that's how brain can represent a lot of different concepts and entities because it's using a ratio of a lot of different activities in order to represent. Yes.
Now, like how to be, now this colors, right, because we have an entire color, like, you know, we have numbers for each color represented, so that we can describe any color according to the combination of RGB.
Yeah.
Yeah, and there's RGB, the C1.
Yeah, so you always get the exact match to the you want by having the number of passes on each screen.
Yeah, so as you said, so if I have, if I have a color bases like red, green, blue, I could create any color from that combination, depending on the ratio of a mix, right? That's exactly this, yes. So it's a ratio system you can imagine that helps me represent something. But I'm gonna press on the word representation. You hear that a lot, right? Oh, how is this represented in the brain? So it's a coding system, right? It's a face in the world does not look like a face in my head. it's just the activation of a lot of cells that is representing that face in my head, so I could connect that face with all the faces I've seen before and then recognize it as being, oh, yes, I've seen this yesterday, or oh, that's a student in my class, right? So everything has to be represented in the brain through those neural activities in order for us to do computations on them.
Just one question, so context is important in that sense for you to activate the right colors, for instance.
Yeah, so you're saying, so for a representation, context makes a big difference, right? And I think I have some examples later on where the way you recognize something changes by the fact that, oh, what is the right context for it, right? So you could recognize, since you brought it up, I'll show you the example. Yeah, so you tell me when you want to take a break. It's 12:15.
Guys, what do you think? We have this room until after lunch, and then we have to change to the small one. So I think we can go, if you're not tired, we can go a little bit more. What do you think, or not? Are you super tired?
What is it, what to lunch, right?
I don't know. So how much time do we have to go? So we can.
It depends on how much we want to get through. I'm looking at it. I'm only on slide 17.
Okay. Okay, so let's go like half an hour or more, something like that, and then we stop. Can you understand? Yeah.
And then we're not meeting again, or what is the
Yeah,
plan?
we are.
Oh, we're meeting later.
Homeless.
Okay,
in
okay.
this building, so we are moving from room to room because today is like just.
Okay, all right, that's fine. That's fine. Let's do it. So you said half an hour, then take a break, then come back somewhere else. All right, that's fine. We don't have to get through the whole thing. I'm just trying to introduce people to some concepts. So this happens, this is something that happens everywhere in the brain. When you perform an action, it's a population of motor neurons that are coding for that action. When you perceive something, when you listen to something, all of these things have to happen as a population of neurons collectively representing that object or that sound or that action. It's just more robust. It's more expressive, right? It has a lot of properties that you would want in the brain. And also it's very graceful in degradation, right? If I lose a color in the example that I gave here, I would lose a person maybe, but if I lose a color here, I still could use the ratio of all the other colors. So imagine I'm using 3 here, but imagine in the brain, if I have millions of neurons that collectively are responding to something, if I lose a bunch of them, it's not going to change my representation abilities. So a big part of NeuroAI is to compare neural systems with AI systems. That's a huge part of it. The reason we want to do that is because when you're dealing with a complex system, there is only one way to understand them, and it's to build one. So for example, if I told you that I have a great model, I have a great understanding of the US political system and social, movements in the US. How would I convince you that that's true? How would I convince you that I have a great model and understanding of the US political system, or, as I said, the social system or social movement in the US? If somebody came to you and told you that, and you would say, no, I don't believe that. What were they supposed to do for you to believe it? If I told you that I have a great understanding of the atmospheric movement and the weather movement and all of those, what would convince you that it's true? What's that? I can't.
You would actually like want to figure it out.
Oh yeah. So let's say you hear it. Would you believe it if I told you, oh, my theory of atmosphere is that whenever, whenever my neighbor has a party, it's likely gonna be sunny. Would you believe that that's a good system?
You're kind of like referring to your own bias.
Yeah, so how would you believe that that's a good, so if, what would convince you that my understanding of this complex phenomenon is actually correct?
It matches your expectations, so if they have an explanation, then.
People already experienced at some point, like you or sort of when your brain has a barbecue or something, then if you hear someone else say it, you're like, oh, maybe that's true, because I have a history or I've seen it for myself.
Yeah, so that's an example of like that's an example of a bad way of verifying something, because then it just matches your own biases, right? But what is an objective way of doing that?
Research,
What's that?
data,
Yes, exactly. So what would these experiments do? What are they testing?
there will be testing whether that result is always present X number of times listed.
Let's say it was present. Let's say every time it rained, every time it was sunny the next day, the day before that, my neighbor had a barbecue. Would you believe that?
The date.
Yeah, but if it's in the past, that's different. It could be just in a statistical anomaly. So
Number of observations.
you could have a million observations and still be in a statistical anomaly, right? If it's all in the past. No, I know what you mean, but you could form an opinion just based on looking at the past that could be purely statistical anomaly or a correlational anomaly. But the way you verify, and the way we verify anything in reality is through testing predictions about future, right? So as I was saying, if I can predict, for example, oh, this team is gonna win, or that this stock gonna do very well, and then in the future it holds, right? A lot of times that's how we know that somebody has a good model of the deeper mechanisms in the system. Does that make sense?
How a person statistical anomality, whereas exactly the prediction also be a statistical anomality.
Sure.
True. So the critique that using past events to a true conclusion would be a statistical anomality could also be applied to predictive power, but even if you predict it correct, then you're wrong.
Yeah. So a lot of time, when let's say if the system is complex, the way to get deeper into that understanding is to build these models, right? You're saying, okay, I use the weather example. If I want to, if I'm building a model of the weather prediction, then the way you would believe that that's a good model is that I could predict the weather in the future, right? I could predict how this hurricane is going to be. The way you develop that model is as you described. You're taking information up to a month ago and then predicting the following day. You're taking information up to a year ago and predicting into the future. So there's always the idea that understanding a complex system would have to come down to, are you able to predict the behavior of the system in the future? The reason I brought that up is that now, let's say in NeuroAI, I so far I said, well, it seems there to be a very interesting cascade of information processing going on in the brain for visual processing. So you start from the retina and the LGN are not shown here, but imagine they feed into V1, primary visual area in the back, and then they cascade forward. And the face area that I showed you is all the way down here, the one that I showed you the recording from. There is an area here called like fusiform face area. If there's a damage to that, people start having difficulty recognizing faces, for example. They get prosopagnosia. So there is a face selective area here. Now, if I have this cascade of information, I can actually record from all of these stages. I can know what the neural activity here was, what the neural activity here was. I showed you some. I can also build a system. For example, we all, in a lot of phones now, we have systems that can detect faces, right? You give it when you're taking a picture, they have these neural networks where they can detect faces at the end of it. They can put a bounding box around the face. Would you believe that the system that does that in your phone does it exactly the same way as the brain does?
Well, you just called that. That's right.
No, I didn't say it does. I said there's a neural network that is trained to detect faces. And it's trained using some methods we're not going to get into. But it's a network. It's in silico. It's in, it's not in the brain. It doesn't have bad material in it. It doesn't have any neurons in it. It's on my computer. It's a program. It's a network. It is a network. It has units in it. I showed you a network earlier, but imagine a more complex. I said ChatGPT is a network. And it does face detection. It can detect faces in the image. What would convince you that that network is a good model of how the brain does it? That's the core question of NeuroAI.
Would, should be human prediction?
Yeah, so there are different ways of testing a model, right? If I have a model and I'm saying this is a model of object recognition in humans, but I implemented it using artificial neural network. So you'll say, okay, does the representation across different layers of this network match the representation across different layers of your artificial system? Does the network detect face as good as the humans do? For example, I showed you how good we are at recognizing faces that are blurred. Does the network do that? So be testing the network behaviorally, but also looking inside the network and seeing different representations in order to see, well, does this representation match what I recorded from the brain or not? So both on the neural level and on a behavioral level, they have to match in order for one to be convinced, yes, this network is a good model of the brain doing this. Does that make sense to everyone? This is the paradigm that people use in NeuroAI in order to build models of the brain, right? You want to understand the brain and hopefully eventually build better intelligent systems through that understanding. Now, I told you what representation is. If I give this picture to the humans and record from some area in their brain, and I have, let's say, 500 neurons, and I look at the activity of them. If I give the same face to the network, I would also have units, and I could record from them. But I cannot really compare those units in the network to the humans. The reason, or to a monkey neural data, the reason is that it could be in different scale, it could be a different system. For example, if you're using RGB to code all the colors, and I'm using, cyan, magenta, and something, I cannot compare them, but we're using the same system. We're using the same basic color system in order to represent all the colors. But I cannot compare my system's RGB to yours. You'll be like, oh, these are completely different systems. But in reality, they're exactly the same system. It's just that they're implemented differently. So that's what makes NeuroAI very difficult, which is, how do you know the principles that are similar are the important ones, and the ones that are not are nuisances? Does that make sense to everyone? Meaning that, I use another example. Let's say you have a ranking of baseball players, and let's say Aaron Judge is top in your ranking, but you use a system that gives 10 to the best and 1 to the worst. So you have this ranking system. And I have a system that gives 1 to the best and 10 to the worst. I cannot directly compare my ranking to yours because those numbers are different. They come from different scales. But if I know that how you compared Aaron Judge to all the other players, so if I have your comparisons in your system, and I have my comparisons in my system, then I could compare those. Because you rank this person better than everyone else, and I rank them better than everyone else, regardless of the numbering system was. Yeah, but we can't, supposedly we could compare ranking, but let's say we wanted to compare the ratio that we have for a specific stimulus. If I want to compare all the ratios, then it would become difficult. So what people, exactly. Exactly. So in order to translate from the representation in the machine to the brain or the other direction, you have to first characterize the representation. That's what I'm trying to say. So what is a representation? Well, representation is that the brain responds, let's say, to this image, to another image, to another image. So I could create something we call the similarity or similarity matrix. It says in the brain, these two faces were very similar. These two were a little less similar. This face and this house were very dissimilar. And this face and that house were even much more dissimilar. You create these constructs in using the brain representation. And you all kind of know what I mean by representation in a way. You could create the same construct in the model. So far, the model and the brain are not talking to each other. You're just characterizing the representation in each of them. And then you could compare those characterization to one another. Again, the baseball example is a good example because if I have a comparison of all the players that shows which one's better than which one, and then I rank those players and be like, okay, I rank this player better than one, this player than that one, worse than this one, worse than that one. If your ranking is the same as mine, we're going to get very similar matrices at that point, even though the numbers that we gave them were different because we're using a different system. And these are called representational similarity analyses that are extremely influential in NeuroAI. So you could do the same thing here. You could take the representation of this figure in different layers in the brain, but also in the network, and then compare them to one another. Be like, am I representing these similarities and dissimilarities similarly in my network versus the brain? And if I do, then maybe that means that this model that I'm using here is a good model of the brain. And that's a really difficult problem to address. Just how one complex system is similar to another complex system is an extremely difficult thing to answer because, again, what matters is context dependent. It's output dependent. There are a lot of things that make that comparison not be trivial. And this is just not that trivial. So let's say we took a network, and I want to give you an example from our own work, where we had a system that across different steps, I'm using you this similarity matrices. And this is a network that is trained for object recognition, for face detection. So we have a huge data set with different faces of, in this case, Dalai Lama and others. And there are, let's say, thousands of identities. Each identity has many images in them. And what we do is that we train the model to separate them, to recognize, well, this picture is Dalai Lama, this picture is someone else, this picture is someone else. Then we could look at these dissimilarity matrices and see that, well, across different steps of this network, pictures of Dalai Lama, so these are not just one picture, there are like, let's say 200 pictures of Dalai Lama. Different pictures of Dalai Lama are becoming more similar to one another versus pictures of Dalai Lama and another identity. So the network is learning, and I'm showing color coding them down here too. The network is learning to make different representations of the same identity be similar. This is akin to what I showed earlier when I showed you, let's say, this representation down here. You're comparing Bill to another picture of Bill, to another picture of Bill in terms of this representation, and then comparing those to different pictures of Samantha, Roger, and Grace, and then drawing that. And what the network is supposed to learn is that, well, at the end, different codes for Bill should be more similar to each other than codes of Bill and Samantha. That's how you get recognition. Recognition arises by the fact that representations, brain activities for one identity become more similar to one. You could also look at how they separate human faces from non-faces. And you see that, well, what they do is that the network's different steps, they actually separate, saying, okay, human faces are, so the difficulty with categorization is that it's very nested, it's hierarchical, right? You could say, well, animate objects are different from inanimate objects. Animate objects have different species in them, humans versus monkeys. Humans have different, categories in them, and eventually they have different identities in them. So categorization is really nested, and one thing could be part of multiple categories, and like Wittgenstein's idea of like, sibling resemblance and those sorts of things. So it makes it really complex. But the idea is that representation should be expressive enough, the sorts of color coding and ratio that I talked about, that allows one identity to be part of many different categories, but also not impossible to categorize them. So that's a difficult problem for the brain to solve. There are areas in the brain that I showed we were recording from, and we could compare the same RDMs that I showed here from the model now to RDMs that we recorded from the brain data, right? We're not comparing brain data and representation here. We're comparing different characterization of them to each other. All right. Maybe you want to take a break here? All right. Sorry, this is...
Nice lunch, coffee, and we come back like in 1:30. What do you think? Okay, and let's go back to the, let's put our things there, so Sean can come here and use the room. I'm sorry, Roseanne,
That's fine.
we'll change